This is the second question of NSEB 2025. If you are here just for the answer, scroll to the end and just check the answer. But if you are here for that short and crisp explanation, let's dig into this question. The question says, which of the following statement best describes the role of, or you can say the function of, ORI site, the origin of replication site, in a recombinant plasmid used in bacterial transformation? That is, in a bacterial transformation experiment, what is the function of ORI site? This is what the question directly has asked. So this is a very basic question from biotech. We have answered this question. We have read this topic a number of times in starting from Stanley Cohen, Herbert Boyer's experiment, components of a plasmid in those parts. So if we start up right here, what is the function of that role of that origin of replication? Say this is a desired gene. This is a desired gene. And what we actually want is to introduce this gene into a cell. This is the host bacterial cell. This is a host cell, and this host is a bacterial cell. And since this is a bacterial cell, say this is its native nucleoid. This is the nucleoid of this bacteria. Now we want to transform, or you can say transfer, this gene into this cell, for which we use a vector. Now this is a plasmid which is to be used as a vector in this experiment. In this plasmid we get a site called the ORI site. This site here, this sequence right here is say called the ORI site. The function of site, this site is, firstly, as the name suggests, this is the origin of replication. How you can say is the normal DNA different from the origin of replication, or how is the ORI site different from the entire sequence of plasmid? It is that the ORI site firstly is an AT rich sequence. This here is an AT rich sequence. That is, if we can say, let's get out of biotech. Let's say this phenomenon is taking place inside a normal cell in our body or in a plant cell. Here an enzyme acts that starts replication is a helicase enzyme. If there is AT rich, we know A and T have double bond within them, two hydrogen bonds, and the other two nucleotides G and C have triple bond within them. So say this one is the helicase enzyme. For the helicase to break or to digest, it is more easier to digest double bond, and it is more tough to digest these GC bonds. So if you see from an enzyme point of view, or say helicase point of view, it is more easier to break the double bonds. So the AT rich sequence is widely regarded as the ORI site. The necessity or the requirement for the origin of replication is that there are more number of AT. Now the number of ORI sites are say 1 in case of prokaryotic cell. That is, in an entire prokaryotic DNA or a plasmid, you will get only one ORI site. But in case of eukaryotes, you will get multiple ORI sites in a single chromosomal DNA. But here we are not concerned about that molecular thing. Here we are strictly concerned about that recombinant plasmid and bacterial transformation. That is strictly from the biotechnologist point of view. This ORI site also decides the copy number. That is, we have a desired gene. Say this is our desired gene. This will make how many copies inside this cell? Say n number of copies are made. This depends on the ORI site. This is called the copy number. Copy number means this desired gene will make how many copies inside. So what would be the answer of this question? Let's check. The first option says the ORI is responsible for antibiotic resistance of a plasmid. No. See, for antibiotic resistance there is something called marker, or you can say selectable marker. For the first option, see, my aim is not only to find the correct answer but also to explain why the other three options are not correct. So this one here, if this one was the question, the answer would have been selectable marker. The function that is given here, antibiotic resistance, these are the functions of selectable marker. Say Here there is a gene called ampicillin resistant gene. Here there is a gene called tetracycline resistant gene. This might be a PBR vector, a plasmid developed by Bolivar and Rodriguez. In that vector, you will get an ampicillin resistant gene and tetracycline resistant gene. They act as marker for identifying the bacterial transformants. But the question says the function of ORI site, right? So let's go to the second option. The second option says the ORI ensures the plasmid is integrated into the bacterial chromosome. See, this is also the function of selectable marker. The first and second, both options, the functions that are given are of selectable marker. Whether the desired gene, see, there is a possibility that this gene has entered. There is also a possibility that it has not entered into the host. Now, how to check? This is checked with the help of marker, something called selectable marker. But the question itself says what is the function of origin of replication. Let's stick to our question. The ORI allows the plasmid to replicate independently in the bacterial cell, correct? The function of ORI site is to replicate. As soon as I see this word, my answer goes right up here. Answer would be option C. Now let's take the fourth option. The fourth option says ORI is involved in transcription of the inserted gene. No. Transcription means DNA. When you use the word transcription, it simply means DNA to RNA. This is what transcription indicates. Now, when you say transcription, it is done by a different enzyme called DNA dependent RNA polymerase. The ORI site starts the process of replication. So here the correct answer would be option C. Thank you.